It's my pleasure being here today talking about this work that was recently accepted for publication just yesterday. And it's very timely because also Dan Turners were presenting this scheme-sec yesterday during his sensational talk during the plenary. So it was a deluxe introduction to the talk. Let's start with the talk, but before we get into more detail, let me present briefly the history of modern animal breeding. Back in the prehistory, livestock was intensive labor with farmers aiming to make crosses to produce more docile animals that can produce food, heat, clothes, and withstand diseases and environmental stressors. During the Industrial Revolution, the human population grew exponentially, demanding amounts of food never before seen. So in the middle 20th century, we came up with these complex mathematical models that take into account pedigree and phenotypes from the farm or testing records to select the best animals to be parents of the next generation, expecting them to pass the best genes on to the progeny. In early 21st century, the sequencing era arrived, and it enabled genome-assisted selection. It is also known as genomic selection. Genomic selection mainly is looking into the genome, so we can be more accurate selecting the animals because we can take a look into their genes. Calculate this polygenic risk score. We call them direct genomic breeding values. And we select the best one for different kind of traits like production, conformation, disease resistance, and also new environmental challenges that come into next years. With genomic selection, we've been more accurate at selecting the animals, and you can see in the plot here that the genetic progress has accelerated a lot. But genomic selection still poses some challenges. The main ones are the number of positions from SNP chips are still limited. Between 10 and 50,000 positions are included in the SNP arrays. It is more difficult to obtain rare variants with SNP chips. Whole genome sequencing is still expensive, so it's not an option. And also epigenetics is getting more and more important to study G/E interaction effects, so we can combine genome and environment, and SNP arrays so far do not provide this information. Genotype-by-sequencing has been proposed as an alternative to SNP chips recently, but we need it to be cheaper. It would be great if they include methylation information and also structural variants, and Oxford Nanopore technologies offer all of them, so it's a great alternative. So after this brief introduction to the history of animal breeding, let me present this story about future animal breeding. And we have include this method here that we have called epigenotyping with low-pass sequencing. We have called it epiclose, and it's similar to the scheme-sec that Dan's presented yesterday. So what is epiclose? It's sequencing the genome, but with a low coverage, so that it's accessible economically for large populations, so we can implement it in the large scale. We need to develop a bioinformatic pipeline for variant and methylation calling using this low coverage, and possibly with some nucleotide miscalling or some error-prone reads. We need then to calculate the merit the genetic merit of individuals using genomic and epigenomic information, rank them, and select the best one to be parents of the next generation. So what we propose is to sequence the genome at a low depth, ideally lower than 2x or 5x, extract SNP variants, also indels and structural variants, impute missing SNPs that have not been genotyped, but we don't have enough information from the low-pass sequencing, and then estimate polygenic risk scores for each animal. Then we need to rank them according to these genomic reading values for reading decisions, and ideally, we could include also epigenetic information. So we were lucky to be in the developer access of this Kit 14, this new chemistry, and the new R10 flow cells. So we tested this method using data from 6 cows. We use blood samples. We extract the DNA using the Monarch kit and fragmented the DNA to 8 kb using these U-tubes. And this table shows a summary of the statistics that we got from our runs. You can see that we got between 5 and 17 gigabases per run, and this is because there was a technical problem during the runs. They stopped all of a sudden, so it could have been even better than this. The N50 of the reads were close to 8 kb, as expected from the fragmentation tubes. And the mean accuracy, as you can see in the table and also in the plot, for the simplex reads were around 98.9%. We got like 30%, around 30% of the reads were duplex, and the duplex mean accuracy was higher than 99%. So this was a very exciting result. This is the bioinformatic pipelines that we developed. I'm not going to get into much detail, but basically we map the reads to the Bostaurus genome, and then we call the SNP variants using CLER3, filtering SNPs using BCF tools to get rid of those variants that with the low coverage are not centered about the polymorphism. Then we need to impute the variants to whole genome sequencing using this 1000 Bull Genomes project. We're lucky to have access to these reference populations so that we can impute the genomes of the animals that are sequenced. And then finally, we estimate the genomic reading values of the individuals and rank them to have to take the decisions. In parallel, we detect methylated regions using Modbam to bed. Dorado, we use Nanopolis for creating the final files format, and then ChipSeeker and DSS to prepare the methylation analysis. Going to the results, you can see here the agreement between the imputed genotypes from EPIC-Lows and the SNP chips, and you can see that we have a high accuracy when we sequence the samples at a coverage of 2x, and even at a coverage as low as 0.5x, we got a decent positive results, but with higher error at the homozygous genotype. So still, the imputation accuracy was dropped to 90, 88, 92%, but still quite good. And this is how the polygenic scores, or the genomic breeding values of the animals, how similar they are to the ones that are calculated from SNP arrays. You can see that sequencing depth as low as 0.5x, we got an accuracy, a correlation with SNP chips of 0.94, so it's quite high. But if we go to 2x, we can have an accuracy of 0.98, so basically the same polygenic score with one method and the other. So low-pass sequencing allows estimating genetic merit or polygenic risk scores with similar accuracy to the traditional estimates that we get from SNP chips, so they are comparable. And what about methylation? Adrian did a good job here. He's sitting over there. We got a good coverage, and even if we have sequenced the genome at a low depth, we had like more than 10 million sites, CPG sites, at a coverage of higher than 4x, and still between 2 and 4 millions of CPG sites sequenced at sequencing and a coverage larger than 10x. So this is quite a lot of data to work with methylation. And as you can see on the plots on your right, there is a quite homogeneous coverage of all the genome with these methylation sites that we cover. Interestingly, most of the methylated regions that we got were in distal intergenic regions. Only 5% of them were close to promoters, so there is a lot of work here to study and to do research with this to know what these methylation marks are doing in other regions that are not the promoters. We compared the methylation from Nanopore and with bisulfite sequencing, and as you can see, the pattern is the same in the genic regions. But bisulfite overestimated the methylation, probably because it cannot differentiate the methylated and the SNP variants, and also probably due to some bias introduced by the bisulfite conversion. Going to the conclusions from my talk, we can say that we can use epiglows to estimate direct polygenic risk score in the populations, do the same that we do the SNP arrays, but with the low-pass sequencing. We could have much more benefits with the low-pass sequencing. The breeding values are comparable to SNP chips, and we can use as an alternative to have more information about epigenetics and so on. SNP variants and methylation status can be discovered simultaneously with no additional cost. We can include epigenetics and GBI interaction information in breeding programs. Epiglows and low-pass sequencing can be implemented in other analyses or fields, GWAS, epiGWAS, plant breeding, and human genomics. And I have a final request to ONT. We need to increase the yield of the chips so that we can have a sample and have genomic variants, methylation, and also microbiota, because we are interested in the microbiota, and we can sequence it all together and develop breeding strategies to increase sustainability and welfare in livestock. Thank you to all the people that collaborated in the project, also to the Nanopore people, and thanks for the support to being here today. Thank you.